Okay, so it's 5 after 2, so why don't we go ahead and get started. So welcome to the CTSI Academy Spotlight Series seminar from the Biostatistics, Epidemiology, and Research Design, or BERD function. Let me figure out how I'm going to change my slides. There we go. So before I get into the designing clinical trials part of the lecture, I just want to remind people about BERD and what we can provide CTSI investigators. Statistical support from BERD is provided through the Zilber School of Public Health Biostats group, and then also the Biostats Consulting Service here at MCW. We provide 4 to 6 hour mini grants of statistical support. The types of proposals that are typically supported include a discussion and review of project aims and study design, review of data and confirmation of data analysis plans, sample size calculation, help with sample size calculation or power calculations, some small preliminary data analyses related to study design and study development, and as well as reviewing data analysis and discussing findings. So you can access those with the link provided here if you are interested in working with one of the BERD investigators or BERD statisticians. Beyond that modest scope of support that you can access directly through BERD, the MCW also has a biostatistical consulting service that can provide more, longer term support and larger project support, and a variety of areas that include data analysis that might not be as easily accessible through CTSI BERD function. To access that, there's a couple of different mechanisms to do that, but we have a fee for service mechanism for either MCW or for outside clients. A number of departments, divisions, or centers across MCW have set up integration contracts so that support is free to investigators because of the support that they provide directly to the Biostatistical Consulting Service. So let's get into the clinical trials part of the talk. So the learning objectives are to understand the phases of clinical trials, become familiar with common designs for each phase and the issues associated with those designs, and learn about the key considerations and inputs for study design and sample size computations. So the typical phases of human research, the ideal clinical trial that you want to get to have a definitive answer about the effectiveness of the treatment is typically a gold standard randomized double-blinded trial. But there are traditionally 4 phases of research to get there. Phase 1 establishes safety and does dose finding and PK studies. Phase 2 is meant to establish biological activity or potential efficacy and explore the potential populations that a treatment should be used in. Phase 3 is that typically that gold standard randomized trial. And then phase 4 involves long-term surveillance in a broader population. And there are certainly a variety of hybrid phases. You often see phase 1 and 2 combined into a single protocol, sometimes phase 2 and 3 as well. So let's talk about each of those areas. So phase 1 trials, the goal typically is to identify how well is the drug tolerated, typically using small numbers of individuals. So this involves toxicity screening and typically a determination of the maximum tolerated dose, or MTD. Sometimes there's a distinction between phase 1A and phase 1B, where phase 1A refers to studying healthy subjects, and phase 1B is disease subjects that are failures on conventional therapy. Phase 1 trials are typically pretty small studies, less than 10 patients per dose. There's often a dose escalation or de-escalation plan. And efficacy may be measured, but it's not the main objective of the phase 1 trial. Really, the main objective is to identify safety or toxicity issues. So what is a maximum tolerated dose? This really is the highest possible dose that has what one might consider an acceptable rate of dose-limiting toxicities. Acceptable may be in the eye of the beholder, but it's an acceptable toxicity rate is something that's set by investigators because of a potential for benefit to patients. So they may be willing to accept a certain amount of toxicities if there is a corresponding efficacy or effectiveness associated with the treatment. Cancer studies, many of these phase 1 trials define an MTD as the dose that produces grade 3 or worse toxicities in no more than 1 in 3 patients. So if there is a dose response or an examination of the dose limiting toxicity rates against the dose level of a treatment, this is typically described through what's called a dose response curve. So the dose response curve plots the likelihood or the probability of a dose limiting toxicity against the dose level of the treatment. And so the point at which, so if we're thinking about 30% as the definition of the maximum tolerated dose, then the point at which the likelihood of a dose limiting toxicity hits 30% would be the maximum tolerated dose. Now you might not have an observed dose at that level. So oftentimes the dose levels are, have an increasing dose levels. A modified Fibonacci sequence is often used, and you can see that here, a doubling of the dose and then a 2/3 increase and then a half, 1/3, and then continuing to go up by 33% each time. But so here, we don't observe the maximum tolerated dose exactly. So the lowest, the next lowest dose would be considered the MTD. One of the more, so I should mention that much of what I'll be talking about and the examples will come from my experience in oncology focused clinical trials. And so some of these may be a little bit more commonly seen in the oncology setting, but certainly many of the features and so forth can be extrapolated to other settings as well. So one of the more common dose finding designs that you see in the literature in phase 1 trials is a 3+3 design. So here, essentially, each dose is considered and analyzed separately. Patients are enrolled in cohorts of 3 for each dose level. And if you have 0 out of 3 dose-limiting toxicities, then you go up. 1 out of 3, well, you're not sure, so you add another 3 patients and try and figure it out. And if you have more than 1 of the first 3 that experience a toxicity, then that's too many, and so you stop and identify the prior dose as the MTD. So if you hit that 1 out of 3 for the first 3 patients at a dose level, and you enroll an additional 3 patients, then you look at how many out of 6 experience a dose-limiting toxicity to decide whether to increase the dose level again or to identify the MTD at the prior dose. So if less than 2 out of the 6 experience a toxicity, then you go up. Otherwise, if 2 or more experience a dose-limiting toxicity, then you would identify the prior dose as the MTD. Typically, you want the MTD to have at least 6 patients treated. So this is an example of what this looks like. So dose 1 has 3 patients. Green means no toxicity, red means a toxicity. So the first 3 patients, no toxicities, you go up. Dose level 2, you've got 1 out of the first 3 patients. So you enroll another 3 and they were all okay. So we're at 1 out of 6, so you go up again. Here again, you had 1 out of 3, so you enroll another 3 and then you would go up. So here is three patients, all okay, so you go up again, 3 patients, all okay, go up again. And here you had 2 toxicities at this 6th dose level. And that is where you've exceeded the MTD. So you go back to dose 5 as the MTD. So the traditional 3+3 design has some drawbacks. It actually tends to underestimate the maximum tolerated dose. And it is trying to target an MTD based on a 33% dose-limiting toxicity rate. That's where you can see that 1 out of 3 and 2 out of 6 cutoff. But even then, it's still underestimating the maximum tolerated dose. Another disadvantage is enrolling cohorts of 3 actually can move quite slowly through the doses, and other approaches would consider enrolling enrolling 1 at a time at different dose levels. The advantage is it's very simple. It's a straightforward algorithm. Anybody can apply it. You don't need to even talk to a statistician to do this. And reviewers and physicians are pretty comfortable with it. There are some other phase 1 designs that are worth describing. So the continual reassessment method is one that is popular. So the idea here is that you actually fit that dose response curve or that dose toxicity curve after each patient's outcome is known. And you figure out where's the corresponding maximum tolerated dose on that curve. And that's where you put the next patient. You put them right at the MTD. And then, when you get their outcome, then you again refit the curve and then put the next patient at the estimated MTD. There are a variety of different modifications to this principle, but in general, it's more flexible. It has better statistical properties than a 3+3 design. You can use it with any target toxicity probability for your MTD. So if you want to target an MTD with a toxicity rate of 20%, then you can easily do that with a continual reassessment method. Another one that is useful is called an MTPI, or Modified Toxicity Probability Interval design. Here you define a target interval or range for the toxicity probability, and then you use the data collected so far at the current dose to to determine the probability of being below, above, or inside that target interval. This is actually a Bayesian approach. And based on that information, you decide whether there are rules that help you decide whether to escalate, de-escalate, or stay at the current dose. And so again, this is a more flexible design than 3+3. It's also simpler than the CRM. You don't have to do any fitting of the dose toxicity curve or anything like that. And it leads to a very straightforward rule-based design that you can show in a table so that investigators can follow it in a straightforward way. And it also can deal with any target toxicity probability. In terms of the sample size of phase 1 trials, the actual sample size of a 3+3 design is driven by what you see. But the maximum sample size that you would typically observe will be 6 times the number of dose levels. That's kind of the worst case scenario where you have to enroll that extra 3 at each dose level. And so you end up with 6 per dose. For other designs, in terms of identifying the sample size, typically the way this is done is to pick some kind of a feasible sample size, acknowledging that these are usually small early phase studies, and then simulation studies are done to evaluate the properties of the design. So they look at, okay, what is our likelihood or probability of selecting the MTD if this is the design that we use? What is the probability of treating patients at the doses that are too high or above the MTD? What is our expected number of toxicities, and so forth? So that's typically what you would be, what investigators would be seeing from a statistician is some kind of a simulation study that describes how the design performs with a particular sample size. So as the sample size goes up, the likelihood of selecting the MTD will typically go up if you've got a good design. So you should be honing in and closing in on that MTD with larger numbers of patients. There is actually software, and I don't know that we need to go into this too much, but some of these designs are implemented in R packages. MD Anderson Biostats group has some software on the CRM design, And there's some online design, building, and simulation software that requires some registration as well. Phase 2 trials is the next step, and here the goal is to determination of if the treatment has any biological activity, estimate the magnitude of that biological activity, and also get a better handle on the rate of adverse events that you might see with this treatment. So, in a study of 6 patients per dose, you are not necessarily going to get a good handle on the adverse event profile. So as you go up to 40 to 50 patients and you've honed in on a particular dose, you get a better picture of the adverse event profile before proceeding to a randomized study. Here you've typically focused in on a single treatment regimen or single dose level, although not always. Sometimes doses and combinations of treatments are being evaluated in phase 2 trials. But the sample sizes are still modest. Oftentimes you'll see them, in the 40 to 50 patient range, sometimes a little bit smaller. And they're used to select the treatment for phase 3 trials. Here you're trying to just confirm that the treatment is doing what we expect it to do. It's biologically active in a way that we expect to be beneficial to patients. Oftentimes the inclusion-exclusion criteria for a phase 2 trial will be a little bit stricter than a phase 3 trial. You're trying to identify a signal, so you may restrict to patients that are most likely to benefit. Typically you're looking for quicker outcome measures if possible. You don't want to, if you're trying to figure out whether to move this forward to a randomized study, you don't want to wait 3 years for the outcome on each patient to be able to make that determination. So here you'll often see what we call surrogate endpoints or short-term endpoints that one might expect to be predictive of a long-term benefit. And then often these are done in 2 stages, and we'll talk about that in a minute. So in terms of the goals, so estimation of outcome is one common goal. Here you're just focusing on constructing confidence intervals for the outcome. Often we're looking at a binary yes, no, success, failure type outcome, which has a success probability P. So if you are trying to establish some kind of a signal for efficacy, oftentimes the phase 2 trial will use a historical control success rate as a benchmark. So if you call that P0, then you can set this up as a hypothesis testing framework. Oh, I apologize for the dog barking in the background. That's my companion in here in the basement where I work. The hypothesis test framework can be set up as a null hypothesis that the success rate or response rate is equal to P0, which is the same as the historical control rate against the alternative that it's better. That's equivalent to whether a confidence interval for the proportion does not contain P0. And so you can, when you frame this as a hypothesis test, your significance level alpha is the likelihood of declaring the treatment promising when P is equal to P0. So that's your false positive rate. Here's an example of a phase 2 trial, again in the oncology setting. This is looking at an autologous transplant for metastatic breast carcinoma. And they had 33 patients with de novo or relapse stage 1 to 3 breast cancer. 2-year progression-free survival is 35%. 2-year overall survival is 78%. They collected toxicity data. There were 2 grade 3 toxicities in these patients. Engraftment data was another secondary endpoint, all patients engrafted. And then to try to figure out if this was beneficial or not, or promising, a historical control benchmark had 29 patients between 1993 to 1997 with 2-year PFS of 17%, 2-year overall survival of 61%. It's not statistically different, but that's not necessarily the benchmark that's used in a phase 2 trial. So in terms of sample size calculations, I will There are some formulas on here. If you have some statistical background, they may make sense. If you don't, they may not make as much sense. I'm not going to dwell on them necessarily a lot, other than to show you that there are ways of doing these sample size calculations. And we will have I believe we'll have a future talk on sample size and power calculations as well. So I encourage you to seek that out if you're interested in knowing more. But briefly, in terms of precision of estimation, the bottom line is that larger sample sizes give you narrower confidence intervals. So one way that you can calibrate the sample size is to figure out, okay, how wide or narrow do we want our confidence interval to be? And then we can calibrate the sample size accordingly. So here you can see that sample size depends on the margin of error or the half width of the confidence interval. That's the plus or minus part. Another way to do it is to think about the hypothesis testing framework, and where the power is the likelihood of declaring the treatment effective when the true proportion is some other value where there's a benefit compared to the historical rate. So larger sample size will tend to yield greater power. And so you can plug in your P0, your historical rate, your targeted rate, your significance level or false positive, acceptable false positive rate, and your power, and there's a formula to calculate it. Here's an example. This is a one-arm phase 2 study with a binary yes/no outcome. So you're looking at event probability or success failure rate versus historical control rate of P0. So this is looking at historically steroids for grade 2 to 4 acute graft versus host disease after a transplant has a complete response probability of 35%. And if we have a new agent that we want to study and we want 80% power to detect an improvement in that complete response rate to 55%, so a 25% absolute improvement with a 5% significance level and a sample size of 41 patients will give us that 80% power. And there's a corresponding rule that says, you would reject that null hypothesis if you have more than 19 out of 41 complete responses. One of the things to consider in a phase 2 trial is whether we should be checking earlier to see if there's evidence of a high complete response probability. So this gets us to the concept of a two-stage design. Here you want to minimize the number of patients treated with an ineffective treatment. So we start with a smaller number of patients in the first stage, let's call it N1. And if we have R1 or fewer successes in those N1 patients, then we stop, declare the treatment not promising, and don't continue enrolling patients. If you do meet that benchmark of more than R1 patients successes in the N1 patients, then you continue enrolling another N minus N1 patients for a total of N. And then at the end, you have a criterion for declaring the treatment promising or potentially successful or not, based on more than R out of N successes. For this to work, you're doing an interim look at the data and trying to figure out if you should stop enrolling or continue enrolling. So this works much better if you've got a short-term primary endpoint. Otherwise, there's a long lag time, and then by the time you follow the patients for the endpoint, your accrual might be almost over. There are online calculators to do this. They typically provide two different options for designs. One of them is called a minimax design, and the other is an optimal design. Minimax provides the trial design with the lowest total sample size. The optimal design provides the lowest expected sample size if the new treatment had the same success probability as the control treatment. So the maximum sample size of the optimal design is actually a little bit higher than the minimax design. And this is what those designs might look like for this scenario. The minimax design has a total of 39 patients, 21 in the first stage, and then you would reject the treatment if you have less than or equal to 8 successes in those 21 patients. Otherwise, you keep going to the 39 patients, and again, reject the treatment as not promising if there are less than or equal to 18 successes. The optimal design is has a little bit bigger total sample size of 44 patients, but it also has an earlier look at the data. So you get to see a check for futility and whether you should be stopping early at an earlier point. They both have the same type 1 error, same power. The expected sample size is actually lower under the optimal design, which is how it's calibrated. This is an example of how the language would look. I'll probably skip over that because I pretty much just stated it. Another important thing to do in phase 2 trials is to do some safety monitoring. So here, you've done some look at the safety signal in the phase 1 trial, but it's still pretty early in the development of the treatment. So you often want to be evaluating the treatment for safety and want to ensure that the frequency of the side effects is not excessive. The sample size is pretty much fixed by the study design for the primary efficacy outcome. But you still want to be able to monitor the safety. So the idea is kind of similar to the two-stage design, except that you actually just want to monitor more frequently. So here's an example, with 39 patients from that minimax design for the complete response rate type study. Suppose we expected or had an acceptable toxicity rate of 10%. And we wanted to monitor after every 5 patients. Well, we could develop a stopping rule or monitoring rule that would recommend stopping if more than 2 out of 5 or 3 out of 10 or 4 out of 15 and so forth patients or toxicity is observed in that many patients. And there are calculators for figuring out how to do this. How to set up those stopping rules as well. And this is an example of what how that toxicity monitoring rule might be described. One of the things to look at here is the likelihood of crossing that boundary during the study as a function of the probability of the adverse event. So if the toxicity rate is 10%, that stopping rule or that monitoring rule was calibrated to have a 5% chance of stopping. Whereas if the toxicity rate is 30%, it has a 90% chance of stopping. So these are what we call operating characteristics of the monitoring rule. It tells you, how does it behave? What happens if the toxicity rate is a certain value? How likely is it to stop? Sometimes you'll see the phase 1-2 designs combined. It may be, they may be just merged together where you do a phase 1 and then you do a phase 2. Sometimes they are, the objectives of the phase 1 and phase 2 parts of the design are actually combined and they're both targeted. So you're trying to find an effective dose that's not toxic. So as a combined goal. And so there are designs that do that as well. They tend to increase or decrease the dose based on the observed toxicity or efficacy outcomes. But the designs are typically complicated, so you'll want to work with a statistician on that kind of thing. So let's talk about phase 3 trials. The design features for phase 3 trials are typically the hypotheses that you're trying to test and the design that you use to test them. So the hypotheses refer to the outcome. What is the outcome that you're hoping that the treatment affects, as well as the type of patients that you're trying to, that you think the treatment will affect that outcome in. The design features that are important are the consideration of a control group and randomization to the control group, so that you get a clear determination of whether the treatment is effective or not. The use of blinding to minimize bias in assessment of the outcome, and ongoing data monitoring, similar to what we've been talking about with safety or safety monitoring or with two-stage designs. So what is the question? So that's the first thing you want to think about when designing a phase 3 trial. That involves 3 components: the treatment comparison, the treatment the treatments that you're comparing, the primary outcome variable that you're comparing them on, and the patient population. At least in a phase 3 trial, there's typically one primary question that is clearly defined and stated in the protocol. There can be a variety of secondary questions, but the primary question is the one where you are devoting your statistical attention, I suppose you could call it that, in terms of being explicit about controlling your type 1 error rate and so forth. So if you ask too many questions, there's a tendency to increase the chance that you find something across all of those questions. So that's why typically they will focus, investigators will focus on one primary question, and then the other ones are considered exploratory questions.
Well, we can ask a quick question.
Yes.
Regarding secondary questions, based on your experience, what would be best to keep that number at? I noticed some of the investigators, have at times many of these. What is your expertise saying in regards to this?
Well, there's a tendency to ask a lot of things, and it's hard to rein that in. To be honest, you're investing a lot of resources and money especially in a phase 3 trial. So you want to learn as much as you can, but you just have to be cautious about what you're learning. These are any of the secondary questions. So you can occasionally spend some of your alpha level or your significance level on secondary questions, but it has to be done explicitly in the protocol, and that elevates it to more of a confirmatory conclusion. But otherwise, you really should be considering all the secondary questions as pretty exploratory. They're more explaining the results of your primary endpoint or generating hypotheses for new studies or things like that. And there's often not a explicit control of the type 1 error rate across all of these secondary questions. So I guess I don't have a good answer for you. Sometimes people will elevate their importance into secondary versus exploratory. That's a little bit semantic because really the secondary are also pretty exploratory. But I don't know. I don't know what to tell you. I think there's a tendency to try and learn as much as you can. But you just need to be cautious about the findings because of the exploratory nature of the secondary questions.
Sure, thank you.
Yep. Dr. Logan, can I ask a question about the previous phase 2 one?
Yeah.
You're talking about stopping rules. So, and you're talking here about expanding your your alpha. So when you're talking about the stopping rules, what is there a way in which you're splitting the alphas there for those different stages in your evaluation, the interim analysis?
Yeah. So are you talking about the two-stage design or the stopping rules for the toxicity monitoring? So
I guess both.
So the two-stage design is focused on futility. So really you're actually not, so typically phase 2 designs don't stop early for efficacy. You don't say, okay, the response rate is great, we're going to stop. You typically will say, okay, the response rate is great, well, let's keep going and gather as much information about these patients. Because they're small studies, they're very early stage studies. So people don't stop early for efficacy. The two-stage design is really meant as a futility stopping rule, where you're saying, okay, we don't have enough responses to this treatment to make it worth continuing. We kind of need to reevaluate or go back to the drawing board or whatever. So from that standpoint, actually, the futility boundary rule, stopping rule doesn't increase your type 1 error rate. So the issue here with the monitoring, sequential monitoring, is that the more times you look at the data, the greater the chance that you see something or that you cross a boundary. So those boundaries have to be generally have to be calibrated carefully. But with a futility boundary, that's actually not an issue. It's because you're not stopping and declaring a success early. You're only stopping and declaring a failure early. And so that doesn't have the same consequences. Now, the toxicity monitoring for the safety, we do have that issue. You don't want to be monitoring for safety and have a very high rate of just finding a safety signal when really it's more because you were looking at the data too often. So, you don't want to kind of artificially increase your chances of finding a safety signal. So these boundaries are calibrated so that they have a particular, what we call type 1 error, a type 1 error rate or chance of crossing the boundary at an acceptable toxicity rate. And that is, this probability is adjusting for the multiple looks. So, the way that these boundary values are actually calibrated so that you cap this probability at 5%, for example.
So that's the family-wise.
That's essentially the family-wise error rate. Yeah. So this is, and the way that they do this is they kind of, well, there's a variety of ways of setting up these stopping rules, and that's probably beyond the scope of this talk, I think. But they kind of have an equal allocation of error rate at each look. This is what's called a Pocock boundary, which gives you the same type 1 error rate used up at each look, but each of those looks has a smaller than 5% error rate. Mm-hmm. I don't know. It's getting a little bit technical, but that's the idea. So this is that 4.7% is across the entire monitoring rule.
Okay.
It's a family-wise error rate. Okay.
Thank you.
All right. Yep. So I think we're on this slide. So the main clinical variable of interest should be the one the primary endpoint should be the main clinical variable of interest that's most likely to be impacted by treatment. Oftentimes, this is a composite endpoint. So, for example, if you're studying heart disease, you could count death from coronary heart disease as well as a non-fatal myocardial infarction. And some of the reason for using a composite endpoint is that you boost the number of events, and the power of a study is often based on the number of the events that you see. So by including multiple types of events, you can boost your power. Those response variables for your primary endpoint should be capable of unbiased assessment. So if not, then you may need to use some kind of blinding so that the person doing that assessment is not aware of the treatment that is being received. And then also, it's you want to have response variables that can be ascertained as completely as possible. You don't want to have a lot of issues with loss to follow-up, missed appointments, and so forth. So if your endpoint requires complicated tests and procedures and so forth, you may have people not showing up for follow-up visits or something like that. So you want to keep that in mind. Here's an example from HIV studies. So you considering possible endpoints, you might consider increases in CD4 counts as a measure of immune function, viral RNA reduction. It looks at the amount of virus in the body. You might look more downstream clinically, time to one or more opportunistic infections. You might look at death from any cause, although that's certainly harder and harder these days, or time to first infection or death. So in terms of the pros and cons, the first two are more biologic endpoints, increase in CD4 counts or viral RNA reduction. These are more of a surrogate outcome. that might be more useful in a phase 2 trial or useful as a secondary endpoint to explain the impact of the treatment. The time to one or more opportunistic infections is looking more at the clinical impact, but the issue here is it actually ignores the competing risk of death without infection. So if a patient dies without an opportunistic infection, you don't know really how to handle that. So this could be useful as a secondary endpoint. Death from any cause may be of ultimate interest, but may not be practical, as patients can have a long expected survival, you know, and especially with a lot of the available treatments these days, that continues to get even better. So a time to first infection or death is a composite endpoint that can be useful. So here, you know, you've got the opportunistic infection component, as well as the competing risk of death included both in the endpoint. Some of the secondary questions that are often of interest, if you've got a composite endpoint, you might look at components of that composite endpoint, some other endpoints. Subgroup analyses are sometimes of interest. Toxicity is important. There may be some ancillary study questions or biologic questions that are of interest. Some of the surrogate outcomes or the early endpoints are often looked at as secondary questions. And we alluded to this with Igley's question about the number of endpoints in the secondary analyses. So the issue here is multiple testing. So the more tests you do, the more the likelihood that some of them are going to be significant by chance alone, even if there's no intervention effect. So the more things that you put in there, the bigger your multiple testing problem is. So that's where you have to be careful about interpretation. Secondary hypotheses are better really at shedding light on the results or on generating new hypotheses, and should be considered more exploratory rather than definitive results. So here's an example of a study from the SWOG looking at post-transplant therapy with IL-2 versus observation after an autologous transplant in patients with relapsed non-Hodgkin's lymphoma with an outcome of progression-free survival. So here we have the treatment, the patient population, and the outcome defined for the study. Secondary questions, looking at overall survival, toxicity, subgroup analyses. They were looking at the interaction between chemosensitivity and outcome, or between histology and outcome. And there were no significant interactions in this particular trial. Let's talk a little bit about the patient population. So the patient population should be defined in advance. You want clearly defined inclusion-exclusion criteria. You know, we talked a little bit about phase 2 versus phase 3. Phase 3, you tend to be a little bit broader in your eligibility as opposed to phase 2. Because you want to think about reproducibility of results and think about who the results apply to. In terms of you know, and the other consideration with phase 3 being a little bit broader is you want the inference to be applicable to future patients. And if you have a very restrictive population, and often you see this, patients volunteering for trials are often different than the general patient population. So if you're restrictive in the eligibility criteria, the results may not apply to a broader population. So that's a consideration as well. From a recruitment standpoint, you're often dealing with an issue of homogeneity versus feasibility. Homogeneity means the patient population is very similar and clear and well-defined and narrow in scope. But feasibility, but oftentimes that may be too restrictive and may be hard to recruit. So from a feasibility standpoint, you may need to broaden the types of patients that you include in the study to be able to enroll and complete the study. So there's a tension between homogeneity and feasibility. You also want to consider potential or high likelihood to benefit. So the concept of enrichment is useful in thinking about this. You want a high event rate. You want the patients that you include in the study to have a high event rate, so that so that it's easier to demonstrate a benefit. So for example, if you have an anti-anginal drug, you don't necessarily want to enroll patients that have only had one angina episode in the last 2 years. You want the patients that have had more frequent angina, so that you are more likely to see a benefit of the drug. So that's the concept of enriching the study population for those patients that are most likely to benefit. You obviously have to weigh the benefits against adverse effects. Pregnant woman is one consideration. Gastric bleeding for anti-inflammatory drugs is another kind of thing. You want to think about competing risks. So you don't want patients to suffer from a competing event, which prevents you from seeing a treatment benefit. So studies of heart disease, if you have patients with cancer in there, the cancer may serve as a competing event and cause mortality that prevents you from following them for heart disease. And then ideally you want them to be able to adhere to the study protocol, although this is certainly a challenge to identify those patients. The design schema is typically recruit, obtain consent, randomize, treatment A or treatment B. So it looks very simple, but oftentimes it's more complicated than it seems in the simple schema. So what does randomization mean? Randomization is the process by which all participants are equally likely to be assigned to either the intervention arm or the control group. The advantage of randomization is that it protects against bias in the treatment allocation and in the selection of patients, and makes the groups similar or comparable on the average, and balances out both known and unknown prognostic factors. Now, you may still see imbalance in any individual study, especially if it's a smaller study on certain characteristics, but that doesn't mean that the randomization was done wrong. It just can happen by chance sometimes. Simple randomization is basically a coin flip, right? You flip it heads or tails. If it's heads, they get A. If it's tails, they get B. Random permuted blocks is done to ensure balance over accrual time. So here the idea is that after every 4 patients, for example, so if you have a block of 4, you have 2 A's and 2 B's in those 4 patients. So the first block of 4 has 2 A's and 2 B's. Next block of 4 has 2 A's and 2 B's. The only thing is that you just randomize the order of those 2 A's and 2 B's within every block of 4 patients. So this ensures that you don't get long sequences of A's in a row or something like that. And then the concept of stratification. So stratification ensures balance across important covariates that we know are associated with outcome. So just with a regular coin flip, you can actually get imbalance on a major variable. So if you want to not ensure that doesn't happen, you stratify, and then you have your permuted blocks within each stratum. So that means that after every 4 high-risk patients, for example, you have 2 A's and 2 B's. And after every 4 low-risk patients, you have 2 A's and 2 B's. Stratification by center is also sometimes done to balance things within a center. Another important randomization issue is the idea of when to randomize. So ideally, you want to randomize as close to the administration of treatment as possible to minimize the dropout. And the reason for that is because the analysis of a randomized trial is typically the standard is to analyze it with the intent to treat principle. And that means that all the eligible patients are analyzed according to the arm that they're randomized to, even if they deviate from the therapy specified in the protocol. So that means if they're randomized to arm A and then they get arm B, they're still analyzed as if they were in arm A. And so if you have a long delay between when you enroll the patient, or when you randomize and when you actually give the treatment, there's more risk that they won't follow the arm that they're randomized to. And that will tend to reduce the potential treatment effect that you observe. The intent to treat principle is done to avoid a bias, or at least a bias in a conservative direction. So if good risk patients decide that the toxicity is not worth a small expected benefit for treatment, they may not go through with the treatment, and then you've got an imbalance in the risk profile of the patients as a result. So the intent to treat does the analysis in a conservative way. So here's an example of deciding when to do the randomization. This is an IL-2 post-transplant study that we talked about already. So registration occurred prior to transplant, but randomization took place between day 28 and day 80 post-transplant. And they had additional eligibility criteria at the time of randomization. So you can see here that 394 patients initially registered, 376 eligible, 194 were randomized. So there's a lot of loss between the time they were initially eligible and when they were actually randomized. And among those that were eligible but not randomized, there were a bunch of refusals, a bunch of toxicities, some progressions. So if you would have randomized at the time of transplant, you would have had a lot of, almost half your patients that would have not gotten the therapy, in a clean way, so to speak. Blinding is important. Patients may experience a psychological benefit of being on a new treatment. The attitude towards the treatment can affect cooperation in the study, compliance with therapy, and so forth. And it can actually affect the response itself. The treatment team should be blinded because they can affect decisions on dose modification, how carefully they examine the patient, when to discontinue treatment, and ancillary care in general. And certainly enthusiasm for the treatment can be expressed to the patient. And then evaluator is important to be blinded as well, because their objectivity can be lost, and this can lead to assessment bias. It's always, it can be challenging to figure out whether blinding is appropriate in a study. The important ethical issue is that there's no undue harm to the patient. But there are a lot of practical issues as well. The treatments need to be similar, dose schedules, similar pills, and so forth. Informed consent is harder, and administration is harder. Sample size. I think we've kind of talked about sample size a little bit already, but the basic concept is that larger sample sizes increase the precision of your treatment effect. And the calculations are based on your primary endpoint of your phase 3 trial. So if we might want 80% power to detect if a new treatment increases 2-year progression-free survival from 40% to 55%. And there are formulas in there, but basically you need to specify your type 1 error, your power, your success rate for your control, and for your intervention. And that's what the formula looks like. So in this particular example, we would need 173 patients per arm. Now, some things to consider if you have loss to follow-up, is everybody going to be evaluated 2 years down the road? So you may need to inflate the sample size for loss to follow-up. There are different methods for different endpoints. I showed a formula for the binary endpoint, but you might have normal outcomes or survival outcomes. With survival in particular, the power depends on the number of events, not the number of patients. There are calculators on the internet, but I strongly encourage you to consult your statistician, and we are always happy to work with you. Data monitoring is important, and we've already talked about this, but basically you want to monitor toxicity and for efficacy or futility. And there's a data safety monitoring committee that typically will look at that. This is an example of the number of looks and how that affects the type 1 error rate. So if you look at the data 10 times, you actually get a type 1 error rate goes from 5% to 19%. So this is why it's important. You have to recalibrate or adjust the boundary or the stopping boundary to make sure that you don't increase the type 1 error. And we've already pretty much talked about that. So I do just want to close with a slide to talk about some recent novel designs that you may come across. So one is the concept of a master protocol. And there's a paper in the New England Journal in 2017 that talks about this. But the basic concept is of a master protocol is that you want to try and evaluate several treatments in more than one patient type or disease within the same trial structure. So there are a couple of variations on this. An umbrella protocol studies multiple therapies for a single disease. So it's multiple treatments one disease. A basket protocol is studying a single therapy for multiple diseases or disease subtypes. So this is the idea that you have a particular treatment that's a targeted treatment going after a particular genetic feature or something like that. And so that feature may be present in multiple diseases. So you may have a basket protocol to look at the treatment effect across these diseases in terms of targeting that particular biomarker. And then a platform trial is kind of a combination of those. It can study multiple therapies for a single disease. It's an extension of an umbrella. It allows you to study multiple therapies for a single disease in a perpetual manner, where those therapies can be discontinued and new therapies can be added in an evolving way. So it's done in kind of a master protocol, and the protocol just keeps getting amended to account for those changes. Another concept that's worth considering is adaptive designs. And this was also published in the New England Journal in 2016. And the basic concept here is to use interim data partway through the study to try and modify your design. So this can include features like increasing your sample size. So if maybe the effect size that you're seeing is not quite as big, but you still want to be able to detect that effect size, you might boost your sample size to be appropriately powered. You might decide that you want to focus on a certain subpopulation partway through the study. Or you might consider dropping arms if you start with a multi-arm trial, say 3 or 4 arms, and then halfway through you drop some of them and just focus on the most interesting ones. These designs are complex to implement both administratively and statistically, so they do require a lot of work with a statistician and with a administrative core that can handle it. But I just wanted to present them just because you may see them in the literature, and they are finding a lot of traction these days. All right, thank you. That's the end of my talk, and I'm open to questions. I know we're a little bit over the 3 o'clock hour, and some of you may need to run off to other things, but I'm happy to take any additional questions. I don't know, and certainly people might have submitted some through the chat, but I can't seem to see the chat. Okay.
Hi, Brent. Thanks for the talk. That was really great. I'm at UWM working in a little bit of different field of biomechanics and human movement and things, but I'm just curious if you can explain maybe a little bit more about adaptive design, because I know that's something that we've kind of looked at doing before, but I didn't have a lot of background information on that.
Yes, that's probably worthy of its own entire talk.
Oh, okay.
But so there, and I would take a look at that New England Journal paper, but because it covers there's a wide variety of adaptive designs. They are used across the entire spectrum of the phases of trials. Mm-hmm. And there's a lot of different ways that you can be adaptive. So in some sense, the early phase trials, like the continual reassessment method, is an adaptive design in a sense, because you are selecting the dose based on the accumulating information. So it's adapting to the accumulating information. And that's really the basic concept, is you let the accumulating information guide further interventions. The implications, there are important implications for whether you're using the adaptive designs, mainly from the standpoint of it can be very hard to control your type 1 error rate. So if you're going to use an adaptive design in a phase 3 trial setting, you have to do it very, very carefully to make sure that you're not inflating the type 1 error rate. Sure. or increasing your false positive rate. Sure. In earlier stage trial designs, you don't necessarily care as much about 5% versus 6% type 1 error rate, or those kinds of things. I mean, there's a lot more room for this kind of adaptation. And so you'll see a lot of things, especially a lot of Bayesian designs that try and to some degree, because they have a natural mechanism for updating information about the parameters. So the types of adaptations that you often see are dropping, most often dropping arms. So you might start with 4 arms in a phase 2 design, and you're just not sure which one is the best. Okay, it can be 4 completely different treatments, and then you might might have a control group, and then so you want to keep the control group, but you want to figure out where where should we place our eggs in this basket among these other 3 treatments, right? So you let the accumulating data. The accumulating data might say, well, arm A looks a lot better than arms B or C, so let's kind of either downweight them. That's one way to do it. So you can actually change the randomization probabilities adaptively, or you just have stopping rules where you just drop arms at, if they meet some kind of a criteria for futility. So that's one common thing that's done. And that's probably one of the more common ones. Another one is a sample size re-estimation. This is probably a little bit more in the phase 3 setting, where companies often like to do this because it allows them to invest a little bit upfront, and then if there's a signal that's somewhat promising but not on the scale that they were originally powered for, they can invest a little bit more and increase the sample size and go from there. It's actually not always statistically efficient to do it that way, but it's appealing to companies oftentimes. So adaptive sample size re-estimation is one where you look at the effect size and halfway through the study and say, okay, well, it's not what we were originally targeting, but it's still worth pursuing. So we just need to boost our sample size and so that we're powered for that. So that's one. Subpopulation selection. So you might have a treatment that is targeting a particular biomarker, but it also might have broader efficacy. So you might start out by enrolling all kinds of all patients or all comers, but have an interim analysis to look at, okay, should we be focusing on the patients that are biomarker positive for the rest of the study, or should we keep all patients in the study? You may see that the signal is stronger in the biomarker positive patients, and so you enroll, you close enrollment to the biomarker negative patients halfway through. So that's a subgroup enrichment decision. Okay. I think those are the main ones. Adaptive randomization is one that's sometimes used. There are people that are in favor of and against the use of that technique, but you certainly do see it in some designs. And I think there's just a lot more flexibility in the early phase setting, phase 1 and phase 2, where you can kind of use the accumulating information and make decisions about what to do next. In a phase 3, you just have to be a lot more careful.
Sure, that makes sense. Yeah, thanks a lot. That's really helpful. Yeah, just touching on the most accepted ones or common ones, I think, for now. Yeah. Yeah.
So to some degree, you can think about a group sequential design where you have stopping rules for efficacy and futility as an adaptive design. I mean, the FDA actually considers it part of an adaptive design class. But there is because you're looking at the interim data and deciding, do we close it for futility? Do we keep going, or do we stop for efficacy? Right. So that it's got an adaptation component in it, whether it's to stop or drop arms, or essentially drop an arm or keep going. So it is considered in the class of adaptive designs, but the adaptation has gotten the types of adaptations that you can do more do now are go beyond that, but they are much more heavily scrutinized by the FDA, for example, in a confirmatory trial setting, because they can impact the type 1 error rate. So I guess I would just say in terms of acceptability, they're broadly acceptable in many cases in an exploratory trial setting, phase 1 and phase 2. Just be very careful about phase 3.
Okay. Thank you.
Yep. Other questions? Oh, I might have some chats here. Let me check. Okay, so there's a question about sharing the slides. So the lecture video, I think, will be posted on the CTSI website. It's for those of you that are interested.
I'll ask one more question.
Yeah, no, go ahead.
So I thought it was, really helpful just to, point out, remind us, that every time you take a look at the data, you're increasing your risk of error. So I was just curious, kind of like, I've always understood that, but never maybe fully understood how doing, trying to answer all the questions for the reasons you said earlier on, of just trying to like get everything out of your data as possible, and how to go about that in a conservative enough way that your your conclusions are still reliable and accurate. So I guess, do you have any like recommendations on that, or in terms of like when you design your study, how to best go about making sure that you don't, overwork your data?
Yeah, I think this is obviously the most crucial in the setting of a confirmatory trial, right? Where you if you're trying to make a definitive statement from the trial results, you want to be explicit about controlling your false positive rate or your type 1 error rate. With the others, there's a little bit of wiggle room, but so really, I would think about it as the biggest issues with the confirmatory trial. So from that standpoint, the most common way to keep that from keep those kinds of things from happening is to focus on a single primary endpoint, right? And pre-specify the analysis. This is what we're going to do. Okay, so from a strictest standpoint, when there's a regulatory agency involved, all the analyses are pre-specified in an analysis plan before the data is looked at. And so, nobody can argue, well, you were mining your data or, things like that. And so, that's kind of the cleanest way to do it. Now, obviously not everything falls to that level of, being reviewed by a regulatory body or anything like that. But, I think generally identifying a single primary endpoint is good practice. Pre-specifying the analysis plan to the degree that you can is a good practice. And that's what the protocol analysis plan will generally tend to do that. Sometimes people will draft a separate SAP or statistical analysis plan that fills in some of the details so that there's no question exactly how this particular model is going to be run or something like that. So that's pre-specification and focusing on a single primary endpoint really are the keys to explicitly controlling that type 1 error rate. Now, when you're getting into the issue of, okay, what do I do with all these other things that I want to look at? Sometimes people will devote some type 1 error rate to those. So you can split your type 1 error rate, 4% for your primary endpoint and 1% to a secondary endpoint or a couple of secondary endpoints. And then you can make, definitive conclusions about those. That's one thing. In general, if you've got 20 secondary endpoints, you might want to consider, and you want to be able to make a stronger statement, you might want to consider, tightening up your type 1 error rate, either a formal multiple testing adjustment or an ad hoc one, where you use 1% instead of 5% or something like that. A Benjamini-Hochberg procedure is something that's commonly done to control false discovery rate, and that can be useful when you have a lot of secondary analyses or hypotheses that you're testing. So, there's no easy way around this. The more times you look, so I mean, and the consequence, the more times you look, the more things you're going to find, unless you adjust for that. And if you adjust for that, you reduce the chances of finding things, right? So you reduce your power. If you have a stringent type 1 error rate and drop that down, then you reduce your power at the same time. So it really depends on whether you're considering an exploratory analysis or a confirmatory analysis. I'd be a lot more careful on the confirmatory analysis side of things. If you're being exploratory, then yes, understand the scope of the magnitude of the number of tests that you're doing, and what that means in terms of the strength of the conclusions that you can make. And are these purely exploratory? Do you have some kind of a multiple testing adjustment so it's a little bit stronger? Those are the kinds of things to think about. It's not an easy thing to. I mean, you could devote an entire lecture or entire class to multiple testing issues, especially with high dimensional data and genetics and all kinds of things. So,
and we got it all, I think. So yeah.
Yes. So, but those are the trade-offs. Think about confirmatory versus exploratory, and cast your conclusions accordingly with those cautionary, with that, with appropriate caution.
Great. Thank you.
Yep.
Thanks, Dr. Logan. Have a good weekend.
Okay. Thank you.
Thank you, Brent.
All right. Thank you, everybody, for attending. And I think we will close it out.
Yeah. Thanks a lot. Bye.
Thank you.
All right. Bye.
Take care.